Hey, good afternoon, everybody. Welcome. It's about time we did a YouTube, I would think. This is one we're going to probably want to keep because many of you that have studied more natural therapies have understood the whole concept that cancer is a consummate sugar consumer. We knew that. And if we go back actually 100 years ago from right now, Otto Warburg, who had a hypothesis 100 years ago called the Warburg hypothesis, which has now been taught in almost every single medical school, every single biochemistry class worldwide. It's 100 years. So I thought I'd share some of that insight so that you could have an understanding. It says the year 2021 marks the 100th anniversary of a fundamental discovery that's taught in every biochemistry textbook. In 1921, German physician Otto Warburg observed that cancer cells harvest energy from glucose in a strangely inefficient manner. Rather than burn it by using oxygen, which is the typical pathway, cancer cells do what yeast do. They ferment it. This oxygen-independent process occurs quickly, but leaves much of the energy in the glucose untapped. Various hypotheses to explain the Warburg effect have been proposed over the years, including the idea that cancer cells have defective mitochondria. We've talked about those before, the little energy subsets inside of every cell in our body. Could it be that there's a flaw in cancer cells, or is it a flaw that's actually to their benefit? They said this defective mitochondria or energy factories, and therefore cancer cells cannot perform the controlled burning of glucose. But none of these explanations has withstood the test of time. Cancer cells, mitochondria work just fine, for example. Thank you. Now a research team from the Sloan Kettering Institute, led by immunologist Dr. Ming Li, offers a new answer. Based on a hefty set of genetic and biochemical experiences and experiments published January 21st in the journal Science. Comes down to a previously unappreciated link between Warburg metabolism and the activity of a powerhouse enzyme in the cell called P13 kinase. You don't need to know that. P13 kinase is a key signaling molecule that functions almost like a commander-in-chief of the cell metabolism, said Dr. Lee. Most of the energy costly cellular events in the cells, including cell division, only occur when P13 kinase gives the cue. As cells shift to Warburg metabolism, the activity of P13 kinase is increased, and in turn, the cell's commitment to divide is strengthened. It's a bit like giving the commander-in-chief a megaphone. Now, this may sound rather basic to you, but when you really think about it, this is the bottom line of why it's so difficult to control cancer. The findings revise the commonly accepted view among biochemists that see metabolism as secondary to cell signaling. They also suggest that targeting metabolism could be an effective way to thwart cancer growth, starve them out. Now, that being said, those of you that have worked with me or that have discussed cancer therapies with me, you know that I've recommended a prep called Avamar Ultra, now called Metatrol, for many, many years. And it's basically what that prep has done when we've used it over the last 10, 12 years. Dr. Lee and his team, including graduate student Kay Zhu, studied Warburg metabolism in immune cells, which also rely on this seemingly inefficient form of metabolism. When immune cells are alerted to the presence of an infection, a certain type called T cells shift from the typical oxygen-burning form of metabolism to Warburg metabolism as they grow in number and ramp up their infection-fighting machinery. So this Warburg theory actually holds even for our own cells. The key switch that controls this shift is an enzyme called lactic dehydrogenase, which is one that we can measure in blood work, LDH. It's made from a response to P13 kinase signaling, As a result of this switch, glucose remains only partially broken down, and the cell's energy currency, called ATP, adenosine triphosphate, is quickly generated in the cell's cytosol. Don't need to know that. Dr. Lee and his team found that in mice, T cells lacking this LDH could not sustain their activity, and as a result could not efficiently fight infections. Dr. Lee and his team said this implied that the metabolic enzyme was controlling the cell's signaling activity. The field has worked under the assumption that metabolism is secondary to the growth factor signaling, Dr. Lee said. In other words, growth factor signaling drives metabolism. Metabolism supports cell growth and proliferation. That's how cancer spreads. So the observation that a metabolic enzyme like LDH could impact growth factor signaling through P13 kinase really caught our attention. Like other kinases, P13 kinase relies on ATP to do its work. So the normal metabolism, the normal consumption that we put in and convert into ATP, feeds the body's natural production of these things. Though the team made their discovery in immune cells, there's other clear parallels to cancer. P13 kinase is a very, very critical kinase in the context of cancer, Dr. Lee said. It's what sends the growth signal for cancer cells to divide, and one of the most overly active signaling pathways in cancer. As with immune cells, cancer cells may employ Warburg metabolism as a way to sustain the activity of the signaling pathway and therefore ensure their continued growth and division. Published in the Journal of Science, it says glycolysis fuels phosphosidase 3 kinase signaling to bolster T cell immunity. You don't need to know that, but it's a fact that Otto Warburg 100 years ago knew what he was saying, still being taught every day in biology or in biochemistry class. And we go on to say that prep that we have utilized, called Avamar, now really comes under the magnifying glass as being something that can do what Otto Warburg said was the way to fight cancer decades ago. Okay? Any other questions about this, or if you want to still set up your own personal consult time, call us at 678-228-8900, 678-228-8900. Our website is thenutritionshop.net. By the way, if you missed our last Friday night first open seminar in Zoom, we will have it up on our website here in the next couple days. So hang in there. You missed a good one, but we're going to do another one real soon. All right, folks, God bless you, and I will see you back here real soon. Take care.